Hello everyone and welcome to today's webinar, High Resolution Imaging of Zebrafish at Nano and Millimeter Scale. I am Jennifer Woods of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Andor. To learn more, visit andor.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Julien Rességuier, Postdoctoral Fellow, University of Oslo, and Dr. Claudia Florindo, Product Specialist, Life Sciences, Andor Technology. Dr. Rességuier, you may now begin your presentation.
Thank you for the very kind introduction. So good morning, everyone. It's a real pleasure to be with you today. And I will present you my work on high-resolution imaging of zebrafish at nano and millimeter scales. So just a few words about me first. So I'm a postdoctoral fellow for the University of Oslo, for the group of Professor Gareth Griffiths, for whom I conduct two research topics, one on the behavior of therapeutic nanoparticles, on the interaction with the vasculature, and especially how they accumulate in disease related to angiogenesis. And also the second topic is more to the characterization of the structural organization of fish mucosal immunity with a special focus on the Gill-associated lymphoid tissue. So now we can start. So basically, in those recent years, there have been many advances in imaging, whether it is new kind of microscopes, such as the light sheets, or great improvements of the confocal microscopes, either conventional or spinning disk. They also benefited from a lot of improvements from the cameras that allow us to acquire data at much higher resolutions and sensitivity. Of course, when you have so much imaging data, you also need improvements of the software. So all these improvements allowed us now scientists to replace the molecular biology data into the spatial context. But the thing is, a lot of the imaging advances benefited a lot the in vitro studies, as you can see with this very nice picture of cell culture with the dragonfly. The problem is, in some fields, there is some kind of a gap between the in vitro studies and the next step, the in vivo. And there is a current need to strengthen this bridge, especially for fields such as nanotechnology and vascular biology, which are complex, about complex 3D structures and environment. And we think that one of the problems with these situations is, well, the mouse model is the most prominent model used at the moment by the scientists, but it shows several limitations for imaging. And we just go past all the practicability things. Still, there's people have been able to have very nice live imaging in mice with intravitreal imaging. However, the problem is this is a very invasive method. So either you just induce inflammatory conditions into your experiment, or you have to put anti-inflammatory drugs. And still, with that, it remains difficult to secure at the same time a very nice high spatial and time resolution. So basically, we have these situations when most people look into in vitro studies on cells and directly switch to in vivo in mammalian systems. And we think that a lot of research would benefit by adding an intermediary model to the mammalian systems, or to use a complementary model to them. And we think that one of the scientific animal model that is on the rise at the moment, and that benefit a lot from all these advances for imaging, is the zebrafish. While it's highly available, accessible, it is incredibly practical because of its small size. And as you can see to the right here, the larvae of the zebrafish are fully developed by the tree, but also they are transparent when you add small drugs. There's a lot of tools that have been developed, and it is ethically more accepted than mammalian systems. But it's not just about practicability. The zebrafish is show high degree of conservation with mammals. 70% of human genes have an autolog in zebrafish. It has a complex immune system, sensitive to toxicity. So actually, a lot of people start to use the zebrafish model for genetics, developmental studies, neurosciences. At the moment, what is put on the rise is human disease models for infections, cancers, or other biological mechanisms. But I think that what makes this model the most appealing at the moment and why the community using zebrafish grow and grow is because of two parameters. It's very good for live imaging and whole organism observations. And the reason why is those two things have everything to do with preservation of the biological context. When you want to address complex scientific questions, you want to have the perfect and most optimal biological context. For live imaging, you don't have fixation artifacts. You can observe live what is happening. You can see the cell moving, what it does. And if you go for whole organism observations, you don't need to dissect things, so you don't do any damage. You can keep all the connections between the organs. If you want to study systemic tissues, vascular, lymphatic systems, nervous system, or even bones, it's very good. And also, to be able to check on the whole organism level, that gives you the opportunity to explore other organs. Like, okay, I want to study the liver, but maybe I would find something unexpected in the organ nearby, in the kidney, for example. You don't know, but this actually allows you to do it. So if we go a bit more into the live imaging now, so as I said, the zebrafish is heavily used for drug screenings, for cancer fields. For example, a group in Portugal by the Professor Rita Fior is injecting cancer cells from patients into the zebrafish larvae, and then she evaluates how they respond to drugs, to chemotherapeutic agents. There's a lot of people working to study host-pathogen interactions on virus, for example, with chikungunya virus, and even now there is preprint about the coronavirus. So by the group of Irene Salinas, Pierre Goudineau, and Jean-Pierre Lebroux. There is a laboratory in Netherlands led by Maria Folenza, that study trypanosome, sleeping sickness in the zebrafish, and then she can actually study how they behave in the blood and infiltrate the tissues. And in our case, we use the zebrafish also to study bacterial infections. We have model of tuberculosis. So if I were to slightly present, so here you have a zebrafish larvae. So you have the head right here on the tail to the right. You can inject the equivalent of the tuberculosis bacteria of fish, which is called Mycobacterium marinum, into the spinal cord, called neural tube at this stage. And see this very thick granuloma in black. And if you were to stain the bacteria here in green and magenta, you see that this area is full of bacteria. And the nice thing with the Dragonfly, so one thing is most of the data I will present you today, the very huge majority comes from the Dragonfly 500, like this video right here. So we have fish where the neutrophils are green fluorescent and the macrophages are red fluorescent. And then you could acquire the whole volume of the granuloma, and then you could follow the behavior of the cells. And you see that there is one here that at one point the macrophage in red will eat a neutrophils, and its cytosol will turn yellow, and then it will die. So see, it starts to be yellow, and then up, it will pop out and die very quickly. There. So that's the advantages of the zebrafish, but it's also very good for other fields like nanoparticle studies. Here, it's a video where we used fluorescent nanoparticles from the laboratory of Matthias Barth in the Netherlands. So basically, you have very tiny nanoparticles, either round or worm-like, and they're incredibly fluorescent. So here, we'll be able actually to see videos, like you can see the particles, but you see that all the fluorescence in white is from the particles. And the nice thing is that it reveals negative all the red blood cells. And if you wonder what all these black things are in the red blood cells, in fish, red blood cells are nucleated, as is the case for some camel species. So just let you enjoy a bit, when you push the Dragonfly to its limits on speed acquisitions, how good is the imaging? You can see the red blood cells are bending and bumping into each other. So I just find it very nice. Anyway, if we continue a bit, I will now present you a bit more like how we could use the Zebrafish model to actually make some findings on the nanoparticle fields and to actually find a new mechanisms at play that actually interact or actually alter the fate of intravenously injected nanoparticles. So it all starts with the Dr. Bernard Verrier in France on his group in the CNRS on its associated startup called Advanced. So what they're doing is basically they created what's called safe by design polymeric nanoparticles. Just to be quick, it's just nanoparticles that are made in a way, in a proactive way to prevent any issues with human health. So for example, they're just made of plain PLA. There is no surfactant. And also all the residual solvents are in accordance with the European Pharmacopoeia. They have a very small size, 200 nanometers, and they are highly stable. And the thing with these particles is they are biodegradable, biocompatible, and bioresorbable. And if we were to look at that's nice nanoparticles, but what do they look? So basically those particles, they are made of interlacing polymer chains, as you can see in these artistic visions. But if we look by electron microscopy technique, so here in scanning electron microscopy and in those two images with negative stain by transmission electron microscopy, they just look like some very tiny ovoid beads. And as you can see to the right, this black dots is actually because in those particles we encapsulated smaller nanoparticles made of gold, which are dense to the electrons. Because that's one thing which is very interesting with nanoparticles is that you can trap compounds in the matrix of the nanoparticles. For example, drugs, adjuvants, or even fluorophore to make the nanoparticle fluorescence. And in addition to that, you can also add some other compounds at the surface. So they use these nanoparticles for different therapeutic applications, from vaccines, mucosal vaccines, drug delivery, and in the recent years, they have been very good at making custom mRNA delivery that they are now doing vaccines and other kind of treatments with it. The questions when I came in, it was like, okay, it works on the therapeutic level, but how can we further optimize the therapeutic applications? One way to do it is actually to try to better understand what are the interactions of the nanoparticle of just the carrier with the organisms on its fate. So the main question, okay, how do they behave in the blood, for example, if you inject it? What would happen? So we add red fluorescent nanoparticles, or we can also have other colors, that we injected in baby zebrafish into the circulations. And then we could actually just see with live imaging, okay, all the nanoparticle goes. So the first thing we did was with a stereo microscope, and you can see the nanoparticle here in magenta, and the endothelial cells in green, because this fish is transgenic. So all the endothelial cells express the green GFP, green fluorescence. But you can see with stereo microscope, the resolution is not very good. It seems to mimic the circulation of the vascular system, but you can't really tell is it associated, is it inside, you don't know. So in this case, we went to the spinning disk confocal microscope. And as you can see here, with just a very nice blood vessel, the resolution is at a new level. It's not comparable. So we then looked, actually, we investigated, okay, several hours after the injections, where would we see the nanoparticles? So in this image here, you have a maximum intensity projections when you would see the vein of the fish on right here, an artery. You can see all the nanoparticles in magenta, so it's associated to it, but then you can just open the stack and go with the optical section. And then you see that the nanoparticles are actually really inside the endothelial cells, not at the surface. Interestingly, so if you look there, the nanoparticle does not mix with the cytosol, which means they are restricted to the endocytic compartment. And other things we could see is that in addition to be internalized by endothelial cells, we could spot some phagocytes in the lumen, also some of them having a tiny bit of GFP. We then wonder, okay, but what's the dynamic of that? How long does it take for the nanoparticle to be internalized and cleared from the blood? So then we made live imaging, or we injected and immediately go to the microscope. And then you can see the nanoparticles circulating. And you see that by around 20 minutes, almost nothing remains in the blood circulation. So this is data we could confirm afterward with electron microscopy. So we know a bit more, okay, what happened on the on the vascular, on the endothelial cells side. But then, okay, so we seem to have some phagocytes in the blood. So we looked at neutrophils first, with green fluorescent neutrophils, and we didn't see anything. So then we looked at macrophages, and we used the transonic line where macrophages are red fluorescent. So in this picture here, you have the vein right here and the artery right over there. The nanoparticles are in cyan, and the macrophages in red fluorescence. And we could recompose with MREs all the 3D structures, all the 3D of the macrophages, and then you could open up and say, okay, here we have some macrophages. Say, yes, we see the nanoparticles, so let me internalize it. So what we know so far is that, okay, the nanoparticles are quickly internalized by endothelial cells on macrophages by 20, 30 minutes. So then we try to look, okay, but what will happen over time? So we tried, instead of using live imaging, in this case, we made like sections through the organism of the larvae. And we looked, okay, here we stand for the nuclei in blue. We have the blood vessels in green and the nanoparticles in magenta. So all the sections to the left are transversal sections this way. So you can see the notochord, you can see the muscles, you have the aorta right here, the network of veins, and then those two rounds here, they are the kidneys, and then you have the developing intestine on the yolk. Right to the right, all these are a longitudinal section through the major vein and artery. So basically what we see that the biodistribution looks very stable. Nothing really happened. The only thing we could see actually is that some clusterization of the nanoparticle around the nuclei, but also at the 24-hour time, we could spot some phagocytes heavily loaded with nanoparticles that we were not seeing at the 30-minute or 4 hours side. Well, that was a bit intriguing. So we look also the same thing with macrophages. So here we look at the main veins, and basically from 10 minutes, 30 minutes, 4 hours, and 24 hours, there's not really changes. They're still inside macrophages. Although it felt that it still looked like the 24 hours seemed that maybe that they may have a bit more, but that's, yeah, maybe it's just the feeling, nothing real. So we continued. I would not present all the data here, but we tried to look and say they go into the vasculature, but could they go into what we call off-target accumulation sites? I want them to go into the blood, but would they reach the brain? Would they reach the liver and other organs? So here we looked at the brain, and we could see that at 30 minutes and 4 hours, the nanoparticles are restricted to the endothelial cells. They do not cross the blood-brain barrier. While at 24 hours, most of them were still restricted to the endothelial cells, we also again started to see phagocytes heavily loaded with nanoparticles. That was a bit strange, and also we could identify that those cells are macrophages or microglia. Okay, so which really seems to have something different with the macrophages over time. So then we tried to figure out, okay, what's the dynamic of internalization by the macrophages? So we actually look between 30 minutes time point and 24 time points at the quantity of nanoparticles inside macrophages. And we could actually see that although there is no more nanoparticles circulating at 30 minutes, the macrophages still internalize nanoparticles. And also we looked by flow cytometry at the fraction of macrophages positive to nanoparticles. So you see like a slight increase from 10 minutes to 30 minutes, but at 30 minutes, nothing is circulating anymore, but still more and more macrophages get positive for the nanoparticles. So, okay, how does the macrophages internalize nanoparticles? They should not be available to them. Like all of it is inside endothelial cells or already in macrophages. So for that, we went with high-resolution live imaging. So we're injecting nanoparticles into the circulations, and then we went to this little area of the zebrafish larvae called the yolk circulation valley, because you already you have very, very thin tissues before the blood compartment. So it is ideal for high-resolution imaging. So basically what is composed, you have like the epidermis, a small basement membrane, a layer of endothelial cells, the blood circulations, on the yolk syncytial layer. And if we're to look in fluorescence, this is how it looks. So here you have in green this layer of endothelial cells. You have some nanoparticles in white, and the macrophages in red. So you have also a lot of macrophages. During live imaging in this area, we could figure out at one point, like we had several set of videos where it seemed that some macrophages would go in contact very close to the endothelium, and at one point we would see a transfer of GFP and nanoparticle from the endothelial cells to the macrophage. But I think you would see it better in the small videos to the right. It will be this macrophage here. So see, it starts to go in contact, like closer and closer, and at one point here, there, takes up a chunk of GFP and nanoparticles. So quickly, we look into the stack of the videos and say, okay, at the moment it's internalized the nanoparticles, the GFP. There's already nanoparticles in the GFP, so they get a chunk of the endothelial cells apparently with nanoparticle inside. But that the nanoparticle inside are still inside endocytic compartments. And we looked a bit after and say, okay, then the macrophage match everything. You see the GFP signal getting lower because it is being quenched by the acidity of the compartment, while some GFP seems to be expelled by the cell. So you say like that was very unexpected. It seemed that just macrophages could just like new bits of macrophages of endothelial cells, and we didn't know this mechanism. It was never reported before. So basically, what we reported in this publication, like, okay, we don't really know what happened there exactly, but we know that at the end, you have the macrophages that internalize in an endocytic compartment GFP that does not match with the fluorophore inside the cytosol of the macrophage with the red. And then you have the nanoparticle inside the endocytic compartment. So if we look into the mechanistic, then it excludes everything which is exocytosis related. Okay, so what could transfer endocytic compartments? Tunneling nanotubes could do it, but in this case, you would have a direct transfer on mix of the two fluorophores, so that does not work out. Well, it could be maybe it was apoptosis-related events, like maybe there was heterosomes from a dying endothelial cells. I say like, yeah, the problem is the donor cell in the videos we have, they stay alive, they don't die, and there's no dying cells around. And then when we looked at the cell death induced by the injection of nanoparticles, beside the mechanical injury of injection, you don't see anything. So it's unlikely to be apoptosis related. But we think what we have to prove that is that it might be related to trogocytosis. Well, initially, trogocytosis was described by exchange of plasma membrane between lymphocytes. But the thing is, no people started to look into macrophages, and it seems that in macrophages they can use, especially with adipocytes, a chunk of adipocytes, the transferred material is then internalized instead of remaining at the plasma membrane. So still, the identification of this mechanism is the topic of further studies, but still like very interesting because it seems that you have the mechanisms that were not expected for the nanoparticles. And it's very important to try to understand, okay, how it works, because maybe we could exploit it for therapeutic applications. For example, to target nanoparticles to the brain macrophages and microglia, which are usually involved in neurodegenerative disease. So that was quite interesting. And then we tried to look also at all these pathways on disease models. So for example, on cancer, if we show here with this publication we published last year, we can actually infect zebrafish larvae with cancer cells, melanoma cells you see in orange in right here. And you see that when we do that, we have a glorious angiogenesis. If we zoom even more, you see here. And in blue, you have quantum dots, just to show that all these vessels are functional. And of course, we also look into the granuloma, the tuberculosis models. But unfortunately, there is no time to present all this. So that's the parts about light imaging. And now it's time to go a bit more into the whole organism observations. So basically, in this case, it's more about the adult zebrafish. So basically, something I do for my research is I will make cryosections through the length of the zebrafish. So you see here with coronal section. And you see that in the block, everything is preserved. We have the ovaries, the intestines, the gills, here you have the head, the tail, here you have the heart. So everything would be preserved. And then you can make some kind of different stainings, for example, with phalloidine to reveal all the actins. and DAPI for the nuclei. On just those two, the pattern of actin staining on nuclei will give you a lot of information to identify which tissue you're in and which cell type you're looking at. And in this case, in the acquisition to the right here, the endothelial cells, the blood vessels, lymphatic vessels, and junctions are in green fluorescence. And then I will just follow the videos. You can see from this 3D multiple field of view acquisition that you have access, you can identify the kidneys and the tubules. Then you can navigate a bit more, and then you can see here this membrane that separates with the liver with melanocyte in black. Then you have all the sinusoids of the liver, and you can see with all this vasculature why it is very dangerous to get injury in the liver. It's mostly blood vessels and hepatocytes. There you can see the ultimobranchial gland, and then you just continue. Then you have some muscles, you will have some bones here, for example. And then you can follow the esophagus on all its vasculature. And then, if you look there, then you would have a nerve that is also preserved, a blood vessel, and the anastomotic vessels around it. So to be able to make real sections, to stain it, and to then use the Dragonfly to make very fast acquisitions of multiple field of view, it's very nice to explore the right areas, and it's very good to look at, say, like just blood vessels, lymphatic vessels, immune system, bones, everything. So here I would, for example, show you. So here we have a fish that is infected with a parasite, fungi, called Microsporidia neurophilia. In fact, the most preferential sites are the hindbrain and the spinal cord. So here you have a corneal section through the fish that is made of 105 field of view of a thickness of 8 microns. So just to say, this huge data took 1 hour to acquire with assuming English. So I can't imagine how long it would have been with a conventional microscope. And then you can see the eyes, of course. Then you have the nostrils to the two sides. When you see there are actually the nerves, the olfactory bulb, the ventral parts of the telencephalon, the midbrain, and the optic tectums right here. Then you go to the hindbrain. Right here you have vertebra, and then you have the spinal cord. And right here all the muscles. And when you then display the immune cells, this is what happens. So of course you see T cells in the mucosal tissues, the cornea, the nostrils, but then you see all the aggregates to the hindbrain and close to the spinal cord. So this you would not see in a healthy fish. So usually they are associated with these parasite infections. But I think the best thing is, again, to illustrate it with video, just to see what resolution you can, the resolution you achieve with such imaging. So here we focus over the nostrils, and you can see all the T cells, individual cells. And then you can move around, go into the olfactory bulb. Yeah, there it is with Star Wars mode. Here you can continue, and then you can see all the difference between the different regions of the brain. You can see all these red cells that you will not see in the forward regions. Then you have the optic tectum, you see some T cells. In the meninges, this is the ear of the fish. Again, with a bit of T cells, some nerves. You can see all the vasculature in the muscles, highly vascularized. And then again, the video will focus on the parts which are infected by this parasite. So you can see all these parts here, they're full of parasites. And what you see here is full of spores. And again, if you continue, you will pass the vertebrae right here. And then you continue to the spinal cord, and then you can also see sac of spores here on all the T cells around. So this is really like triggers infiltration of immune cells in the brain. So you have to be careful when you do neurosciences to not have your fish infected with these parasites. Okay, so now, there's a few one of the video, I forgot about it. So yeah, we could also spot some local infiltrations of lymphocytes in the brain right here. So okay, now let's go to the parts. Say one of my research topic is to look actually into the immune system of the fish and its organization. So yes, the zebrafish model is heavily used to study immunity, fish and human pathologies, immune mechanisms, but still, there is still a huge need to better understand the anatomical organization of the adult zebrafish immune system. And if I were to summarize a bit, so here you have a very nice zebrafish, and you have the head with the dorsal cavity, and then you have the kidneys, thymus, and spleen. So there is two primary lymphoid organs, the thymus and the kidney marrow. Kidney marrow, this is where you have the hematopoiesis. But the kidney marrow is also a site of, has also a function of secondary lymphoid tissue, which also have the spleen. Then, of course, we can consider that the fish is a huge mucosa because all its interface with the environment is made of mucosal tissues. So you have lymphoid tissue associated to those mucosae, like in the nostrils, the gills, the guts, and the skin. And of course, you can have inductive structures such as melanomacrophage centers and granulomas. The thing is, if you look into these mucosal tissues, what is perceived wrongly is that it's just like diffuse, unorganized, or scattered immune cells in those mucosal tissue, which is a bit odd thinking when you think about the fish in its natural habitat, heavily exposed to pathogens. You would expect the mucosal tissue to be a bit more sophisticated. On the tree, there has been some more structured, now that are being more structured lymphoid tissue that are now being discovered. That will go a bit. So, but my main area of interest is the bronchial cavity. So basically, if you look at the fish from the back, you would see the pharynx in green, for example, in the middle, and the bronchial cavity would make two cavities on each side. And then you would have 4 pairs of gill arches. The water would be breathed through the mouth, goes into the pharynx, reach each bronchial cavity, irrigate the gills, and be expelled by openings. If we look at what gills actually look like, so well, here I change a bit because this is gills from salmon. It's a bit bigger, it's a bit more easy to show and explain how the gill are structured. So basically, at the bottom, you have a supportive structure called the gill arch, very cartilaginous. And then from this gill arch, you have some kind of trees that arise on each side. So if you make transversal section for this, you see like the gill arch, and then you have these two trees called filament. And that in between the filament, you have another supportive structure called the septum. And when I spoke about having structured lymphoid tissue, here you have one actually that has been found in Norway in salmon first, and that we recently showed existed in zebrafish as well. It's called the interbronchial lymphoid tissue that we just call ILT, that is on top of the septum and along the inner side of the filament. Of course, if we want to look into zebrafish, so this is zebrafish gills. So here you will have a bit of the pharynx in the middle. Then you can recognize the gill arch. So this little spine here are called gill striker. They are to prevent bigger food particles to be into the gills. Then you have these small trees called filaments. And then you see that these little flaps to the side, they're called lamellae, and this is where the gas exchange occurs. And if we were to make, again, a transversal section through the gills, because we have this study we wanted to look at the organization of the zebrafish gill-associated lymphoid tissue. So we take this kind of imaging, and again, we have phalloidin in green and dapi in blue, and we label T cells in red. And if we take into consideration the labeling of T cells and the compartmentalization of the gills, we could find that the GALT could be divided in 5 sections. One is this interbronchial lymphoid tissue you see on top of the septum right here, so on top of it, in the side of the filament. Then there is 3 regions, like in the other part of the filament and the bronchial arc. There, the T cells are just scattered, but then something interesting is we could see on the sides, each side of the gill arch, right here on here, at the base of the filament, something that looks a bit more clustered. So it's a new lymphoid tissue that we found and called the antibronchial lymphoid tissue, or ALT in short. So we tried to characterize that a bit more, and you can see with a further zoom, so you have 2 different gill arch. You can see very well the scattered T cells on the gill arch, and then when you reach to this place, you see that the T cells are a lot more clustered. So then we wondered, okay, but this is just some kind of loose tissue, loose aggregate of T cells, or is it structured? So what we did is we labeled cytokeratin, because cytokeratin will then reveal the reticulated epithelial cells that you usually find in structured lymphoid tissue. So you find them in the ILT, which was shown in salmon, and in this new lymphoid tissue, the ALT, we could find that it was the same. So this seems to be like a structured lymphoid tissue. If we look at this small scheme, so basically we have the Gill arch, the two filaments, lamellae, ALT, and then you would have on the two sides the ALT. But still we wondered, is it some kind of aggregates that we find sometimes, or is it continuous for the whole length of the Gill arch? So in this case, I had to dissect very carefully to not damage the gills, which are very fragile, and make a one-month staining with Z70. And then again, that I acquired with the Dragonfly, and you can see that this is continuous. You see everything there, all the T cells in white were continuous. So you can see all the gill racers, the gill arch, the filaments on the flaps. There you have the T cells. You see it continuous. This is the ALT. I'll just let you enjoy the videos just to look at the 3D of the gill arch. Then we could see that, you see, it goes along the side, but then it makes small penetrations in between the filaments. So it seems also to zigzag a bit in between the filaments, which is quite interesting. And as you can see, that the resolution is very, very good. This is very quick to acquire. I think it takes maybe like 5-8 minutes maximum for 3 different colors. So we could find actually the organization of the Gill-associated lymphoid tissue, find a new structured lymphoid tissue in it, and then we wonder, okay, but what if we actually change the level and actually look at the whole bronchial cavity level? So there, actually, we did the same thing as I showed you in the previous videos. We have these multiple field of view acquisitions on transversal section through the bronchial cavity. And as you can see, on the roof of the bronchial cavity, you have the thymus. Here you have the T cells are in white, the actin is in green, and nuclei in blue. And as you can see, we could see that there was some kind of continuity of the T cell staining from the thymus to the gills, actually that most of the parts in the bronchial cavity were interconnected together. But the main surprise that we got is when we then started to look at the lower parts of the bronchial cavity, we actually could find a new lymphoid organ that we call the Nemosian lymphoid organ. So if we just look in this image, the T cells are in red, phalloidin in green, and DAPI in blue. You have the two bronchial cavity. You have the gill arches, 1 and 2, 1 and 2. Then you have the uroial bone right here. And then we see all these structures heavily loaded with T cells, actually in the isthmus, in the part between the two bronchial cavity that connects. And we could see that in many different places. So of course, we characterize that a lot more on the paper. It's very close to submission now. But we could use that, for example, with a collaboration with Montpellier that make layer by layer imaging, focal microscope, merged with vibratomes. We could actually recompose the 3D structure of this new organ. So this is how the Nemosian lymphoid organ looks. Actually, if you were to replace with the heads of the fish on the bronchial cavity, so here you have the two thymus on top, this is the localizations right in the middle of this new lymphoid organ. So you can see the zebrafish just allowed us with live imaging to find new biological mechanisms regarding nanoparticles. And then just to explore the immune system of the fish, we could find new lymphoid structures on new lymphoid organs. So I would like to thank you for attending this show, and also to acknowledge Gareth Griffiths, my PI, to allow me to have the freedom to do the research that I like, all the different collaborators involved in the different projects, and of course Andor and Lagroup staff, as well as the imaging facility Normic that I'm been using and from which I get a great support. So yeah, If you have any questions, so the question and answer sessions will be at the end of the talk of Claudia. And yeah, I'm going to turn it over to Claudia. So yeah, it's all yours.
So thank you very much, Julien, and thank you so much for your excellent presentation. I did enjoy it quite a lot, seeing multiple uses of the zebrafish. And now I will follow up with a very quick overview of the Dragonfly, which was the microscope that Julien used to do most of his a very nice microscopy data. So as you saw from Julien's presentation, it is extremely important to integrate the cells, also the nanostructures within the cells, and to integrate all that information in the whole organism. And we at Andor, we do believe that we have the perfect system that allows you to analyze the nano information, the structures on the cells, and also the whole organism. So starting by comparing the Dragonfly with single-point scanning confocals. So as you can see immediately in this image, when you're using point scanning confocals, the image is scanned point by point by point. And the Dragonfly, due to its multi-point confocal technology, will deliver an image in an instant. And that's why we say we have an instant confocal. Also, because of its dual microlens system, we have high efficiency in capturing the light. Not only that, but the Dragonfly benefits from Andor's EMCCDs and SCMOS technology, which delivers quantum efficiencies up to 90%, whereas point scanning confocals, the quantum efficiencies are around 45%. So we have better efficiency in capturing the light. So as an overall key specifications of the Dragonfly, we deliver a very fast confocal with speeds in confocal mode up to 400 frames per second. It has a very wide field of view with a 22mm diagonal, so this will increase your productivity because you need to acquire less images to have this larger fields of view. And we deliver a very wide excitation range of wavelengths from the 405 nearly 800 nanometers excitation, and it has a very flat, uniform spectrum across the whole excitation wavelength. And our detectors deliver 16-bit gray levels, more than 65,000 gray levels. So the Dragonfly multi-point confocal is what we say a multimodal, high-contrast multimodal platform. As I just told you, it is a confocal, an instant confocal, multi-point confocal, but you can also use the Dragonfly to have laser wide field imaging, or you can also have super resolution techniques such as DStorm and SurfStream, or even you can use the Dragonfly for turf imaging, and all of this in a single imaging system. As I just told you, you have from visible to the near infrared wavelengths with the Dragonfly. And now I will pass to show you some imaging applications that you can use with the Dragonfly. So you can use the Dragonfly for deep imaging applications, and you can image up to millimeters deep in tissues, or you can image deep in tissues and simultaneously acquire live imaging data, or acquire live imaging data and simultaneously acquire double color labeling. And if you combine it with SurfStream, you can acquire super resolution images that break the diffraction limit of light far beyond the edge of the cover slip. So one example of this is this clear tissue that is 2.4 millimeters depth, imaged with the Andor Dragonfly. And because we deliver a very broad, uniform illumination spectrum due to our Borealis patent illumination delivery, you can see that with some systems you can actually see the image, the tiles when you do the image, but using the Borealis perfect illumination, you don't see the tiles. So you have seamless stitching, perfect illumination across the whole field of view. And what does this do? That if you want to image a full fish, as Julien was just saying, imaging the zebrafish, but in this case, for example, I could be interested in the eye, in imaging the details on the eye, but I also want to see the whole fish. So I can acquire with a high magnification lens, and I can acquire multiple tiles because the system is quite fast, and I have the detail at every scale, from the nanometer till the millimeter. And for example, in this case, where we image the neurons, and this is in the mouse brain tissue, and you can see this was more than 5,500 images acquired in 25 minutes. So you have montage and montage with deep imaging, and you can see the neuron and the neuron integrated with its neighboring cells. Moving on to live imaging. In live imaging, the greatest challenges actually is to avoid phototoxicity, because in the end of the movie, you want to have the cells alive, and you don't want that what you are seeing is effect due to degradation and due to too much light. But also, when you go over the phototoxicity, you also want to avoid photobleaching, because in the end of the movie, you still want to have signal enough in order to be able to analyze your data. And many times, researchers need to compromise, need to decrease the light, need to decrease the temporal resolution or the spatial resolution. But because we deliver a system that is extremely gentle in imaging and is extremely efficient in capturing the light, we do believe that with the Dragonfly, you don't need to compromise. And so I will show you some examples. As for here, we were imaging live inside the intestine, imaging deep the blood flow, and this was acquired at 200 frames per second with Andor SCMOS camera, and you can actually see the blood flow. If you want to break the resolution limit, you can use SurfStream. SurfStream yields an increase in resolution between 2 to 6 times in the final data, and due to its low power requirements, it is compatible with live cell imaging. And SurfStream algorithm allows acquisition of super resolved images deep inside the cells and tissues. And you can compare here the same fixed cells, in this case, where I wanted to show the improvement in resolution from no surf to a SurfStream image. Another application that you can use the Dragonfly for is multiplex imaging or spatial result transcriptomics. Spatial result transcriptomics or multiplex imaging is a group of methods that deliver information at the single molecule RNA level of expression of numerous genes. So these methods are extremely important because they allow the discovery of new biomarkers, identify new cell types in responses to diseases and injuries. They can reveal an atlas of gene expression of a 2D or 3D tissue, and they can identify temporal and spatial gene expression patterns. And how this is done? Well, this is done integrating the Dragonfly imaging system with a microfluidic devices and imaging your cells, imaging your sample, doing barcodes and doing multiple rounds of hybridization and stripping, and then with the Dragonfly ultra-fast imaging system doing multiple rounds until you have rounds enough to deliver the number of gene products that you want to analyze. We do offer at Andor a full complete imaging system from acquisition to analysis. And upon acquiring your imaging data, you can go to Imari's workstation and analyze your imaging data. And in this case, for example, trace your vessels. So the Dragonfly comes in two models. We do offer the Dragonfly 500 and the Dragonfly 200. The Dragonfly 500 is the complete imaging system in which you have all the imaging modalities as TERF, the super resolution, and also it can be combined with Andor's photostimulation devices. For example, if you combine it with Mosaic for optogenetics to analyze in cancer research, cell proliferation, and cell invasion, or in neurosciences, for example, you can use TERF to analyze neurocone dynamics and forming connections. The Dragonfly 200 is offered as a more compact solution and is offered in upright and inverted systems. You can use it for developmental biology studies. For example, you can also combine it with Andor photostimulation devices such as micropoint for laser ablation. You can use it for cardiovascular studies, for vascular damage and repair. Again, you can combine it with Andor photostimulation device or for neurosciences. These are very versatile devices, that microscopes that you can use for multiple applications. And this is just an example of multiple publications that have been used with Dragonfly. So with the Dragonfly, we are very proud to say that researchers like Julien do publish science in high impact journals, and we are extremely proud to deliver and to help those researchers to push science further. And with this, I will finish, and I'm happy to take any questions.
Thank you
very much for listening.
Thank you, Dr. Rességuier and Dr. Florindo, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, very impressive imaging. Can this also be extended to study pathogens?
Very good questions. I mean, definitely, there's a lot of lab now that are using the Zebrafish to study host pathogen interaction, from different viruses, different bacteria, parasites, even the coronavirus. There is no preprints that are available, and I think that the paper will be published very soon. So it is, yes, a very good system to study pathogens and diseases.
Okay, our next question is, let's see, where did it go? Great talk. I have two questions. One, can you target the nanoparticles to a specific type of cell and deliver drugs specifically to, let's say, peripheral neurons? And two, what software imaging system was used for the T-cell infiltration after viral infection video?
Well, thank you for the compliment. Regarding the targeting of nanoparticles, that's a bit of a tricky question because it really depends on which way you want to administer the nanoparticles, how long you want them to impact, and also which part of the body. And for peripheral neurons, then you have to think, if you inject it intravenously, you have to think of a strategy so that they would cross the blood, the endotheliums, the basement membrane, and then reach these peripheral neurons. I think that's that's very tricky to answer. I will go to the second part of the question. So I think you refer to the very large brain images with the infection with the Microsporia neurophilia. So for that, the software was Imaris, and well, you make the videos with Imaris, and you try to crop to work with it, and then you can end up a bit with imaging. But just to say, this set of image was more than 100 gigabytes, so you need also a very good computer to be able to work on it.
Thank you, Julien. And we have another question here. We have been using zebrafish larvae for mycobacterial infections. Any idea if the nanoparticle imaging can be used for bacterial infections?
Yes, actually, well, some data I did not present, but we also work on delivering new therapeutic compounds to granuloma, tuberculosis granuloma. So definitely you can do that. And actually the laboratory of Gareth Griffiths has been publishing two recent papers. One that showed that macrophages could internalize nanoparticles and migrate to the site of Mycobacterium granuloma. And also that if you inject it intravenously, they would also accumulate at the granuloma site over time. So you can check the literature of Gareth Griffiths and you would find those two papers.
Dr. Florindo, do you have anything that you would like to add? Okay, I'm not sure if I can hear Dr. Florindo, but we will definitely come back to you if you have anything that you need to say. I will move on to the next question. Stunning pictures. I am working on an osteoporosis. Is that you, Dr. Florindo?
No.
No, okay. I am working on an osteoporosis model of zebrafish. What is your recommendation imaging technologies for drug screening using scale?
I would go for, well, scale are quite thin in zebrafish, so you should be able to do 3D imaging using the Dragonfly very well. You could also use some, well, I don't think you need to go with the light shift. I think it's not big enough, but that could also be a possibility. But then, if you were to use just regular bones for osteoporosis, like all this 3D imaging, if you take the fish, you fix it, cryoprotect it, and decalcify it with EDTA, you can also do these whole-organism sections and try to look over a different set of bones, all the impacts of the different drugs. I hope that answers your question.
Okay, thank you, Julien. Dr. Florindo, are you still there?
I am still here. Can you listen to me?
I can hear you now. Yes, yes.
Finally.
If there's something that you would like to say that we passed over, please go ahead and say that.
Yeah, I was when Julien was talking about the image, so I was just wanting to say that we do offer with the Dragonfly that Julien used for acquisition. We do offer an end-to-end solution, and the image comes integrated with the Dragonfly system for analysis and creating those virtual movies that you have been seeing in the presentation. And sorry for the technical problems.
That's okay. I think we're all kind of used to technical problems now with this year of being on Zoom and webinars, and so I think we all understand. I'm so glad you got a chance to speak. Okay, so let me ask another question here. We may have time for just a few more. Can we get XYZ coordinates information from Dragonfly?
Yes. So the Dragonfly is a multimodal system that is delivered by Andor Technology, and you have it's a multi-point spinning disk confocal. So it is a confocal imaging system, but it also delivers other imaging technologies. So you can do confocal, you can do wide field imaging, you can do surf stream with the Dragonfly, you can do turf, and you can do storm and 3D storm. We do have two different types of the Dragonfly 500 and 200. So if you want to have more information with the Dragonfly, we'll be happy to come back to you offline with the Andor Technologies.
Thank you so much, Claudia. We've got another last question. I think this is also for you. For multiplex imaging, how is the interaction between the Dragonfly and the external devices achieved?
So thank you so much for that question. And for the multiplex imaging, the interaction is done by the REST API interface, which is incorporated into the Fusion software. So the external devices can be controlled by an external program that can be driven by Python, LabVIEW, or MATLAB, and this provides the ability for Fusion to communicate for Dragonfly to communicate with the microfluidic devices through Fusion and to proceed to the multiple rounds of imaging and washing, integrating with the microfluidic system.
Thank you, Dr. Rességuier and Dr. Florindo. Do you have any final comments for our audience today?
So, Julien, please go ahead.
Oh, okay. Well, thanks for attending the webinar. Also, thanks a lot to Dr. Florindo for the invitations to perform this seminar. I hope that after these presentations, you would be convinced of how powerful is the Zebrafish model for live imaging. that you would also consider all the great improvements that have been done on the spinning disk microscope, on that all the things that you can achieve with it. So yeah, that was my final words.
So as for my final words, the first big thank you I would like to give is for Dr. Julien, who was kind enough to present his work and these awesome images. And thank you, Labrut, for giving us the chance to present our system. And I would like to say to everyone that we do have an outstanding imaging system, a spinning disk confocal microscope that allows us to acquire confocal images quite fast and deliver explained resolution. So please contact Andor if you want to know more about the Dragonfly. And this is all from my side.
Thank you again, Dr. Resseguier and Dr. Florindo, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Andor, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.